back to the Peace Offering podcast. And today we're interviewing Dr. Robert Thompson, who's a longevity and anti aging physician in Alaska. He has two offices there, one in Anchorage, and has been writing and researching, especially in the field of minerals, over the last 30 years. It's pretty impressive. Most of this research done with hair tissue mineral analysis or a hair test looking at mineral composition. But we get into talking about calcium, potassium, iron, and iron toxicity, which we've covered before, but he's got a completely novel way of looking at it, as well as the importance of whole food vitamin C. I hope you enjoy this interview as much as I did. So, how I found you was I brought Morley up for an event. We did a conference, 3 weeks ago. Feels like a year ago. It's 3 weeks ago. And during that time, I said, "Hey, who should I talk to?" And he brought up your name and then forwarded me a really interesting thread that you were both going back and forth about. Anyway, what really piqued my interest was you were talking about fibrinogen and how it being a sponge for iron. And so that was one of the things that I wanted to talk to you about. You got back to me about HTMA. And so I'd love to hear about how you utilize that. And, if there's anything else that you want to talk about with regard to metabolic health or longevity, we can certainly get into that. And if there's anything specific you want to cover, we can do that as well.
Definitely. All great subjects that I love to talk about. I have a
Okay.
whole bunch of books I'm working on right now on the subjects. Right now, we're trying to finish the calcium Y3, and I have several new concepts I'm introducing in there. Uh, Most significantly is that osteoporosis I had previously determined was a nutritional disease, but I've now determined that it's exacerbated by dilution. And I'm like, dang, I had no idea. And I did it to myself, ended up with a broken back because of it. And I'm like, I wish I'd learned on somebody else first, but I got to learn on myself. And I basically have been incorporating hair tissue analysis into my practice for over 23 years. And I quite simply had the worst decline in my tissue mineral levels I'd ever seen in any patient ever.
Hmm.
I had 30 to 50% drops in all my mineral levels in my body within about 16 months, and I'd never seen that before. And I kept trying to figure out why, and it ended up I was drinking about 4 to 6 bottles of molecular hydrogen water every day. I have a little hydrogen generators. I sell them. They're fantastic. It's kind of cool. Remember, molecular hydrogen is our molecule of energy of mitochondria. Anyway, in the process, I diluted all the minerals in my body and then ended up suffering osteoporotic fracture. And I'm like, dang, Robert, you wrote the book. What the heck did you do wrong? And I realized it was all about dilution. And then I realized this was the reason, this is the reason that my patients haven't been improving more over the past 23 years in my practice of medicine with incorporation of hair tissue analysis because we're still diluting, all of us. So basically, I've been saying for 23 years, 25 years now, actually, the two most important things we put in our body every day are water and minerals. Our bodies are 72% water, 28% minerals on average, tissue wise. And so I thought to myself, how come you weren't taking minerals every time you were drinking those water bottles? And I realized I diluted my minerals by doing that. How many people you know drink lots of water?
My answer is it depends. It depends on because I have my fingers in a few different like nutritional communities, and
Well,
in some of those they're advocating lots of water, and some of those are advocating very little water, and but the ones that are advocating very little water are the ones that are talking about minerals. And
two most important things we put in our body every day are water and minerals. We're 72% mineral water, 28% minerals in our tissues in our body on average. And so if we're drinking and we're not taking minerals, we're diluting those minerals that we have. And so all osteoporosis is a disease of nutritional deficiency and it's all exacerbated by dilution. And so we have to get in the habit of taking minerals, trace minerals, every time we drink, every glass we drink, no exceptions. So I put the minerals in my car, I put them in my kitchen, I put them in my bathroom, I put them in my bedroom, bathroom, every place I could, next to my computer at my office, every place I could possibly drink, I have trace minerals handy. So I can't possibly dilute without, taking more trace minerals. And it's a foundational principle. It's in my new book, the Calcium Y3 coming out. It changes the history of the world. It really does. No one's ever recognized this before, and it's too simple. But no one's been measuring hair tissue analysis for as long as I have, and making these revelations. So the second thing that I teach would now I think it's just foundational is potassium, all about the most important of all minerals to human physiology. Do you know what that is?
Well, if I understand you correctly, it's potassium, and I'm assuming this is
Oh,
because of the calcium potassium regulation in extracellular, intracellularly.
well, let me tell you more about it.
Yeah.
It's going to blow your circuits in the big picture, and it's all basic science. It's not opinion. And I tell doctors right to their face now, either you listen to what I'm saying or retire. Because if you don't, you're reharming patients 90% of the time. It's completely unacceptable. Okay, so potassium is the most important of all minerals to human physiology, and 94% of all the potassium in the human body is inside the cell. So we can only measure it by hair tissue mineral analysis. That's it, no other way. Blood tests are only 6%. Why would you measure 6% when you can measure 94%? I call the 6% the stupid doctor test. I mean, seriously, blood tests for minerals are stupid doctor tests. 94% of all the minerals in the human body are inside the cell. You must use hair to measure them. All patients, all the time, no exceptions ever, because potassium is the most important of all minerals to human physiology, and the only way you can measure it's hair. And the real big question becomes why? Why is potassium so important? And by the way, this is in Dr. Pollock book, Gerald Pollack's book, Cells, Gels, and the Engines of Life, 2001 publication, pages 119 to 122. Look it up, doctors. It's pivotal to the history of the world. So potassium is the highest in concentration by molality, and the why is dipoles. Dipoles. Do you know what a dipole is?
Yeah, vaguely, but go ahead and explain for the listeners. Yeah.
Two poles. Two poles. Two poles. What has two poles? A battery. So this becomes critical, really, really critical. Potassium is required, and it's attached by ATP. Two statements. You have to have potassium, you have to have ATP to our protein molecules at every other peptide, the carbon chain. On that carbon peptide, every other carbon is positive or negative, and it's 7 angstroms. And so on the negative carbon, potassium's attached by ATP by a cool mechanism that I figured out, because you got a triple negative phosphate attaching a potassium. How's it going to do that? When it attaches the potassium, electrostatically, the potassium attracts water molecules up to 600 atoms thick. That's quite literally what hydrates the human body. So without potassium, you have no water in your cells. None, zero. You can't hold on to it. It's not there. Your whole body's completely dehydrated. You have to know your potassium. Every single person has to know their intracellular potassium. I tell doctors, start with yourselves. It's the most important of all tests.
So can I just jump in here and ask you, if I another way of say maybe addressing this other than hair tissue, would be to do a bioimpedance test on your intracellular water. Are you saying it's a requirement to have potassium to fully hydrate the cell to have it
Yes,
the
yes. You have,
so
you
would
have,
you see
you don't have,
lower intracellular water as a result of a potassium deficiency?
remember, water is attached to our protein molecules by potassium. All cellular hydration depends on potassium. all. And that's why when we cut a cell into two pieces, it doesn't run because of potassium is attracting the water molecules electrostatically. So it acts like a gel instead of a liquid. Okay, so why is this so critical? Because it's not just all cellular hydration. What do the batteries do? This is what really blew my circuits, and because I never heard anybody talk about it before. And I did this lecture in November 4th, 2023, and I told the doctors, I call it the Nobel speech, because in the history of the world, the entire world has missed it. The entire world. Albert Centauri, Dr. Warburg in 1931 got the Nobel Prize for describing the physiologic changes in the cell that lead to cancer, basically anaerobic glycolysis. Everybody missed it. In 1972, Dr. Centauri wrote his book, The Living State. In there, he talks a lot about the energy of the cell. In 2014, Dr. Seyfried from Boston College published his book, Cancer of Metabolic Disease. Well, the interesting thing about that is he basically reiterated everything Dr. Warburg said. But all these brainiacs have missed one half of all the energy of the cell. One half. Without measuring potassium, you have no idea. It's called direct current energy. What does it do? What's it responsible for? I've never heard anybody talk about it, ever. In all the
Yeah,
years I'm
well, so so you're referring to like the work of Robert Becker. You're familiar with the body electric. He was a Yale professor. He did
yeah,
work on salamanders and found the DC current was what regenerated the limbs of a salamander, et cetera, et cetera.
yes, but this is us. This is our human body, the most important mineral, and no one's measuring it. Nobody understands what it does. Dipoles, dipoles. So it turns out that remember we have prion proteins. When proteins are shrunken and misshapen, they don't have water on them, and they have a calcium excess. Calcium's a +2 ion, so it can stick those two proteins together without water and create what we call zippers. And that's really a big problem in cellular energy because all of a sudden all these proteins are misshapen and they're dysfunctional. They're called prion proteins. We actually have scientists that study these and they believe that they're associated with disease, but I think it's a step previous. It has to do with the energy. Okay, because all DNA compaction, histones, all DNA replication, nucleosomes, all all protein synthesis, all RNA synthesis depends on this direct current energy. The average person that I test has about 15% of what they're supposed to have. That's average.
Hmm.
And no one's measuring it.
How
No
do you
one.
measure it?
Hair tissue analysis. That's it.
Okay.
That's the only way you can measure it. Seriously, that's it. That's why I tell all the doctors right to their face, do hair tissue analysis on all your patients or retire because you're harming patients 90% of the time. It's not okay. Okay, so I'm going to make some more powerful statements as we go on here, but this is very, very significant. So calcium causes zippers, the fourth thing I teach. Number one, dilution. Number two, potassium, most important mineral. Number three, dipoles, and dipoles are so critical for all nuclear function of the cell. It's like, dang, no wonder we have so much cancer when we only have 15% of the cellular energy that we're supposed to have. And by the way, thyroid hormone can't work without potassium. You don't have potassium, you're severely hypothyroid, and the doctors aren't doing the proper tests. They're not making the proper measurements, and they're using the TSH test, which has 23 articles that show it's not reliable. At what point are you going to start listening to your own literature, doctors? Seriously, it's ridiculous. It's beyond ridiculous. I'm a teacher, and I'm like, I can't make it up. How many articles do you need before you quit relying on the stupid ass TSH test? I'm serious. You have to be stupid. You got 23 articles that say it's not reliable, and they don't even know why. I mean, I try to tell them sometimes, and they don't listen. So, for instance, synthetic T4 suppresses TSH 400 to 700% more than natural T4. So, if you have a normal TSH and you're on synthetic T4, what's the chances you're hypothyroid? I did the study. I'm almost up to 2,000 patients. It's 1 in 500 chance you're adequately treated with synthetic T4. 1 in 500. And this is cellular energy. It depends on potassium. Thyroid can't work without potassium. Nobody's measuring it. Dang, doctors. Okay, so calcium forms zippers because it's a +2. And calcium doesn't need ATP to attach itself to those two proteins and zip them together. And this is actually the principle of muscle contraction with actin and myosin, calcium and zippers. And you need sodium to pop those zippers. So 90% of all salt restriction is harming patients. Harming. It's just not okay. It's wrong. They're not doing the proper measurements. They make the wrong recommendations. They make the wrong conclusions, and they give the wrong treatments 90% of the time by not knowing the most important of all minerals to human physiology. And by the way, I'm up to 3 years asking now doctors. I've yet to find one doctor that knew the answer to that question. What's the most important of all minerals to human physiology? You'd think a doctor might know that, right? Dang, talk about important. But remember, I didn't know that till I read Dr. Pollack's book in 2021, and it was published in 2001. And I'm like, what the heck's the matter with me? How come I didn't know this? So I claim to be the chief ignoramus on all this stuff when I teach. But I'm still like embarrassed because of the standpoint I'm like some enigma in rural northern Alaska doctor. I'm not at some university someplace where, I have a PhD and all kinds of students studying from me, but I teach those guys. And so the key is we have to understand that we can't pop the zippers without salt. And so we have to know what we need. If somebody's restricting their salt and they need it, they're harming themselves, right? 90% of all hypertension is caused by calcium, not sodium. Zippers makes everything stiff and hard. It's pretty obvious. And the fact that they're not paying attention to what I'm teaching is not okay anymore. This Thompson Dobriner principle that I teach is foundational to the human and to the entire animal kingdom and all disease. So these substitutions occur and they cause problems always. So I've told the world, and I'm telling them again, autism is 100% preventable. If we have one more patient with autism, they should be suing the OBGYN doctor for not recommending iodine during pregnancy. Iodine completely eliminates the effects of bromine and fluoride on the developing brain in the second trimester. You have to get at least 12.5 milligrams. We know that increases babies' IQs over 12.8 or 13.2 points in two large studies in 18 states. Why the heck aren't we recommending iodine in pregnancy? What the heck's the matter with the OBGYN doctors? And why do they have this stupid iodophobia? Where did that come from? I looked at all those reactions and all those things. They're so temporary. If like you get those basalt gastric um, stuff, it's all temporary. It's just a TSH release, probably caused by bromine, causes increased temporary and thyroid hormone production. You might feel it, but it goes away quickly.
What form are you referring to? Potassium iodide?
Actually, yeah, I like glucose because it's a combination of sodium and potassium iodide, and most people need both sodium and potassium. But on hair tissue analysis, if you have a high potassium, you might want to switch to sodium iodide if you have just to minimize that. But remember, almost all atrial fibrillation is caused by low intracellular potassium. You think this is not a problem? 45%. I talked to a cardiologist. He said, Robert, 45% of my practice is atrial fibrillation. I'm like, dang, it's completely preventable. So is autism. 40% of breast, prostate, pancreatic, thyroid, and ovarian epithelial cancer is completely preventable by taking iodine, 12.5 milligrams a day, 40% reduction. I hear these commercials for black people wanting to all of a sudden reduce cancer because it's higher in black people. They want to reduce cancer. Their goal is the next 5 years, reduce cancer by, I think they said 50%. Well, they can already reduce it 40% by just putting everybody on iodine. We can already increase babies' IQs by 12.8 or 13.2 points by just recommending iodine. We can eliminate autism. Why are we not doing it?
if taking potassium iodide, would that disassociate so that the potassium that is joined to the iodine in that form be able to be used intracellularly?
Yes. What
You kill
doctors
two birds with one stone.
well, yeah, what doctors don't realize is every single hormone in the human is iodized. Every single one, not just thyroid, cortisol, estrogen, progesterone, everything. So when you get bromine, which is put in our flour and bread products, into your body, and it goes to those places where iodine is supposed to be, it messes up your hormones. So then you get cysts, you get dysfunction, you get PMS, you get hormones not working because basically of dysfunction from bromine. So this actually was all documented in the 1920s when they went to table salt, and everybody, all these children got cretinism, and then they added iodine to the table salt, and the cretinism went away. But they forgot that they should have been using sea salt all the time, which is 1.8% minerals. And so that helps everything. The fifth thing I teach, which you got into earlier with regards to Morley, is iron and iron physiology. Okay, so it's a really big deal. So iron, quite simply, is the reason that humans die. So if I ask you what causes a disease, so I say, what causes dementia? Your answer should be iron. What causes cancer? Iron. What causes type 2 diabetes? Iron. What causes macular degeneration? Iron. What causes all infections in the human? Iron. Why are we dying? Iron. And guess what? Men don't have menstrual periods or babies, so they all have to be blood donors or do therapeutic phlebotomy. There's no exceptions. Um, Patrick, what's his name? Can't remember. He wrote a book, Dumping Iron, in 2015.
Yeah.
Brent,
Yeah,
Brent
PD Mangan.
Petey Mangan. Yeah, it's a
Yes.
really important observations in there. And I call it slide 53 of my lecture, that 300% reduction in lung cancer, 150%, I'm sorry, colorectal cancer, of 150% reduction in lung cancer, 90% reduction in cardiovascular disease, 60% reduction in incidence of cancer, 40% reduction in death of cancer, and 20% reduction in stroke, just from being a blood donor as a man, because you don't have menstrual periods or babies. And so the formula for men is age minus 12 times 2. So if you're 50 years old, you should have donated blood 76 times. And so yeah, you have to start thinking about that. Here's the key. We can't measure iron, doctor. We can't measure it. There's no test. All the blood tests that they offer from the labs for measuring iron are fictitious at
Okay. All
best.
right. Okay. Okay. So let me just slow you down a little bit because there's a lot coming through your stream right now, and it's all amazing, but I want to make sure I get this right. Let's bookmark the conversation about HTMA because I want to ask you more about interpreting that data
Yeah.
because there's a lot there. But this formula that you just gave us, age minus 12 times 2, you're saying that's the amount of units that you should have donated in order to be basic to essentially nullify the negative effects of iron. So,
That's
I just did the math here, and my number is 72 units. Well, obviously that I can't, one cannot go about that uh, immediately. So, what is a good frequency? So, we're talking particularly about males right now, but how do you judge the frequency of phlebotomy or the frequency of donation that a male could do, especially if they're, say, over the age of 40?
Well, that's a great question, and unfortunately, blood banks regulate the frequency of taking blood
Well,
with,
let's forget about them for a second
with
and
most
say,
parts. So
you're ideal.
they only do it every 8 weeks, and so, um, but the interesting thing is that there's a 5% chance in our population that patients will have hemochromatosis. And according to the literature, hemochromatosis, you're supposed to be homozygous in order to have the disease, and that's wrong. It's completely wrong, 100% wrong. Hemochromatosis is inherited by incomplete penetrance. So the textbooks of autosomal recessive inheritance are completely wrong. I got the book, and there's 4 chapters. I tried to memorize it, but it's kind of bogus. Anyway, the key is that it's an incomplete penetrance. So what's that mean? Well, the gene turns on or off randomly in utero and depends on how many genes turn on or how many don't, whether or not the disease expresses itself and to what level it does. So this is why patients with one gene for hemochromatosis can have really high iron levels and high ferritin level. However, ferritin is not a measurement of iron. However, when we have too much iron, sometimes ferritin can be very high. Back to the whole thing with fibrinogen. So ferritin is a ferring protein. It moves the iron out of the cell, out of the mitochondria, and it's only produced when the body doesn't have the C molecule. So that's scurvy. So we're actually dying of scurvy, all of us. We're dying of chronic scurvy. We're dying of iron over a lifetime. However, we know we die in 3 months or less from lack of C molecules. However, in 1934, the Hoffman-LaRoche Drug Company began producing ascorbic acid and called it vitamin C. Did that make it a vitamin? No. Dr. Signori told LA Times on March 13th, 1937, that ascorbic acid was useless. I'm saying, as Morley is, ascorbic acid is harmful to the human, harmful, because we lack the enzyme to break it down called guanylactone oxidase. We don't have it. So it's toxic to us. And so it increases the destruction of a specific enzyme, ceruloplasmin, loses its copper, and that enzyme is necessary for converting used iron in our mitochondria as Fe2 to Fe3 for incorporation into ferritin. The C molecule is responsible for recycling the Fe2 used iron back to Fe2 plus back to Fe2, so it can be reincorporated into the FES cluster for reuse. That statement is historically significant. No one has ever put all that together before.
No,
And I
and
tell
I think it's
doctors,
going to take a second time of you
yeah,
stating it for it
I
to
tell
land.
doctors too, if you repeat this information, you must give credit to me. Don't be thinking that you invented this knowledge, okay? Well, this set of knowledge that I'm presenting is unique to me. And so just give me credit. That's all I ask. Okay, so the ceruloplasmin is destroyed by ascorbic acid. The C molecule recycles the used iron back to Fe2 for reincorporation into FES clusters. And those clusters can occur in 4, 6, or 8 configurations. So they're always cubed. They're completely square molecules. What's a square molecule doing in the cell? I mean, I try to wrap my head around some of this stuff that I discover and try to figure out, okay, what's that all about? Okay, so but we have FES clusters in a whole bunch of enzymes. For instance, xanthine oxidase has 2 FES clusters in it, and so this whole thing of utilizing iron and keeping iron reduced is a really big deal. And back to the spike protein disease. So remember, I described this spike protein disease in November 4th, 2021, very early in the course of the whole dogma, whatever's going on. In March of 2020, it was published that spike protein attaches to the beta 1 N terminal domain. That's really specific. So hemoglobin molecules have 4 pieces, beta 1, beta 2, alpha 1, alpha 2. And each molecule is connected. And each molecule has a carbon end and a nitrogen end, each molecule protein. And on the nitrogen end, spike protein attaches to beta 1 hemoglobin. That's so specific, it's unbelievable. The world has missed it. They keep thinking that they're focusing on the ACE receptor and all that kind of stuff, and why it affects humans. Completely missed it. And so all the physiology of the harm of the spike protein disease, whether that be infection or post-vaccination or being shed on, you got the disease, spike protein disease. Your cells are producing spike protein. Spike protein attaches to beta 1 heme, so you're destroying heme molecules. That's a problem. It's an insidious death because you lose heme molecules. Eventually, you can't make heme molecules. You need mitochondria to make heme molecules. If you can't make mitochondria, mitophagy fails. That's a problem. And I'm beginning to find patients now where they can attach potassium. Remember, I said you had to have ATP to attach potassium to pro. They can't attach it. I put them on huge doses of potassium, and 6 months, a year later, there's no change in their intracellular potassium levels. I'm concerned about their ATP production, big time, big time, big time. So remember, there's a teeter-totter of energy in the cell. Dr. Warburg talked about it. When that teeter-totter goes the wrong way, all of a sudden you got anaerobic glycolysis. That's cancer cells that don't respond to the normal turnoff of cellular functions. And I think that it all comes back to this nuclear energy, which is pretty much related to potassium. So we must start measuring everyone's potassium correctly. We must begin to understand the significance of iron and its toxicity, the C molecule and reducing its toxicity. Avoid ascorbic acid. Don't ever eat any frozen fruits or vegetables ever. Not ever. Not
Mm.
ever. Not ever. If you're doing smoothies and you're putting frozen berries into your smoothie, you're screwing yourself because you're getting ascorbic acid. The maximum density of water is 39.1 degrees, one of the other things I teach. So if you freeze the C molecule, you get ice molecules. Ice molecules are atomic knives. They literally, it's a flat molecule. It cuts everything to pieces, completely to pieces. So this is why when you eat frozen fruits or vegetables, they taste sour or bitter because you cut up the hexuronic acid off of the C molecule. The C molecule is about 17% hexuronic acid. The other name for hexuronic acid is ascorbic acid by Dr. Senori in 1933 in Scientific American. He's the one that first described it in that name form. But he later told the world it was useless. So it's like, dang, it's still being propagated today. And I tell doctors, I tell my colleagues, I have a lot of integrated colleagues that are doing intravenous vitamin C. I tell them, stop it. You're harming patients. Stop it. Now, why do people feel better after they get that? Well, it's a biome. Okay, so you have bacteria in your body, you suppress it. Ascorbic acid is like an antibiotic. You have an energy boost, but the problem is you're destroying mitochondria. And so that's terminal. You can only have so many mitochondria. You keep destroying them, pretty soon there's an energy problem in your cells, right? And so we want to keep doing everything to keep our mitochondria as best we can, keep them healthy.
Okay. So I don't know that I got a clear answer from you about how one would, let's like forget about blood donation. Let's just talk about phlebotomy. How would a doctor or even a practitioner that had like was how would they
Back to the,
measure whether or not the rate at which they can de-iron themselves?
that's a really great question. I'm sorry I didn't answer more specifically. I tend to go by the ferritin level. A patient has a ferritin level over 100. I like to take the blood even as often as every 4 weeks. Once it's below 100, every 8 weeks is okay. Ferritin is the most important of all blood tests to human physiology. It's the only blood test that's directly related to all death and disease. The only one. Cholesterol is 50%. Flip a coin. How accurate is it? 50%. Flip a coin. Ferritin is directly related to all death and disease. For every 100 points your ferritin goes up, all death and disease is increased 10 to 15%.
Okay,
I
so let
talked with
me
hematologists at LabCorp and I told them this information. I gave them the references and nothing changed. They still offer these iron tests and they still consider up to like 250 normal for ferritin. That's absurd. Patients get results back and it's in the middle of the results and they think they're okay. They're screwed. They have increased death and disease 10 to 15% already because their ferritin is above 100. There's no measurement of iron. Low ferritin is not a measurement of iron. Don't recommend iron, doctor, for low ferritin. Low ferritin is really good.
okay, so if ferritin so if okay, so what I think I hear you saying is ferritin is a good marker for all-cause mortality, and that you want it, you definitely want you start taking action when it's over 100, but you also want it as low as possible. And so here's a question I have not been able to get an answer to, and that is if 70% of of iron is in the form of hemoglobin, and then you have 10% stored in myoglobin, and then you have another, what, 10% in ferritin. Is there any danger for in taking your ferritin all the way down to zero if your hemoglobin is optimal and your serum iron is optimal and your transferrin is optimal?
Those are all fictitious results. Transferritin, all that stuff is all fictitious. Um, it's all fictitious. You got to remember that when fibrinogen absorbs amyloid, I'm sorry, when fibrinogen absorbs iron, it shrinks in coils. At 30% coiling, it's considered alpha amyloid, a prion protein of fibrinogen. At 40% coiling, absorbing more iron, it clumps and folds on itself and becomes insoluble beta amyloid. So, amyloid is the storage protein for iron, which is why we can't measure iron, and which is why low ferritin is not low iron because we can't measure it. It's an amyloid.
Okay, so you're presenting a sort of an alternative way of looking how iron is stored to what I'm, familiar with, which is kind of the RCP way of looking at it. And, it's also hard when you kind of look at the iron biology books, like what the hell's going on there. But are you saying that one could measure fibrinogen and get a sense of your iron dysfunction?
Well, that's a really fantastic and intuitive question, actually, because as I studied severe spike protein disease, we're talking severe COVID. Um, I One of my most fascinating patients was a hemochromatosis patient, and he was heterozygous. He'd been doing monthly blood donations every month for over 12 months prior to getting COVID. And I told him, when you get COVID, you need to come in right away because the problem is that you're going to get a lot of free iron release, and that's going to make your blood really thick. And so um, he came in day two, the second day of COVID illness. We couldn't get his blood to flow through catheters. And I managed to manually suck some out, send it for analysis. And within 24 hours, his hematocrit had increased from his normal around 48 to 64. And what does that mean? Well, what happened was spike protein attached to his beta 1 heme released all that iron, and it caused the body to respond to a lack of ability to carry oxygen. And so he got a left shift in his hemoglobin curve. He massively produced a huge amounts of red blood cells, made his blood count high. What else happened? Well, because he released all that iron, what's the sponge for iron in the blood? Fibrinogen. So his fibrinogen went up to about 700, and his ferritin level went up to,
After the donation?
no, before. This
Oh,
is this
okay. So fibrinogen was at 700 before the phlebotomy.
before the phlebotomy,
Got it.
and his ferritin level was around 14 or 1500. Yeah, so basically all this iron is released. It's got to get soaked up by the fibrinogen. It becomes amyloid, and we can't measure it. But could fibrinogen be an indirect measurement of free iron? Maybe, but it's acute. Um, I had a similar problem in a pregnant patient, and her fibrinogen level was around 700, and her ferritin level was around 1100, and she would have died if I hadn't treated her.
So when you say acute, what time scale are you talking about?
I'm sorry.
When you say acute, fibrinogen could be a measure of free iron, but it would be acute, what time scale are you talking about when you say acute?
Well, probably 24, 48 hours. It's the fibrinogen is going to come back down because it's going to change to alpha amyloid eventually, and then you're not going to be able to measure it anymore.
And so you can't. So right now, like LabCorp does not offer alpha amyloid blood test or
No, there's
beta amyloid blood tests.
no blood test for amyloid. And it gets deposited in the tissues a lot. The other interesting aspect of it, Morley brought this to my attention a few years ago, is that ascorbic acid in the neuron, the neuron of the brain doesn't produce ferritin. Very, very interesting cell. It also has a calcium concentration 200 times lower than all the other cells in the human body. It also has its own calcium channel blocker. But why is all this so significant? Well, ascorbic acid is a sugar molecule. It uses the glucose receptor to get into the cell. Some people try to say it's got its own receptor. I don't see any reason for its own receptor. It makes no sense. So the glucose receptor brings it into the cell. It's identical to glucose within about 9 molecular weight difference. So it has one OH. Ascorbic acid is glucose with one OH. And so we have to realize that in the neuron, if you get ascorbic acid, it gets absorbed and it's going to destroy ceruloplasmin. Well, what's the problem with that is you can't recycle iron without ceruloplasmin. Can't get it into this out of the mitochondria without mitochondrial transferritin in the neuron. So the neuron mitochondria produces mitochondrial transferritin. Well, guess what the stimulation for amyloid formation in the neuron is? Mitochondrial transferritin. So it carries it out and simulates the formation of amyloid in the neuron and deposits the iron. Really? So everyone taking their Centrum Silver? Really? Everyone taking their ascorbic acid big doses? Really? I mean, seriously, do they really know what they're doing? I have a guy goes to my church. He's like taking like 3 grams of ascorbic acid every day. I tried to tell him, and he won't listen to me because he claims Linus Pauling did. And I'm like, well, yeah, but Linus Pauling died of prostate cancer, and ascorbic acid didn't help him. His wife died of terminal pneumonia. Guess what the cause of death is from scurvy? Terminal pneumonia. Most of our seniors die of terminal infections. Why? Too much iron. Iron's built up in their tissues. Antibiotics don't work anymore.
I'm still working through your explanation of your this new pathway of fibrinogen to alpha amyloid to beta amyloid to prions, which is really remarkable because what that is, eh, like the math that I would do doesn't apply anymore because you just introduced a whole new variable. So in your opinion, what percentage of iron is in hemoglobin?
Well,
Or what percentage of stored iron is in hemoglobin versus all of the other potential places?
that's another really complicated question. And historically, the textbooks have suggested 70%. But I don't know because we have over 100 hemoglobin molecules within the human. All the cytochrome enzymes are hemoglobin molecules. 4 of the 5 enzymes of the mitochondrial electron chain transport are hemoglobin molecules. I mean, bilirubin, biliverdin, myoglobin, haptoglobin, uh, xanthine oxidase, all these are hemoglobin molecules. All your cytochrome enzymes, over 60 of them, almost all of them are hemoglobin molecules. So the hemoglobin molecule is designed to transfer electrons more efficiently. That's what it's supposed to do. But that's the problem is with the spike attaching to the beta 1 heme, the configuration, you get free iron release. And so what we want to do is balance that as best we can. So we want to get rid of iron, reduce its toxicity, keep the ferritin as low as we can, support our immune response as best we can, because all this stuff is being destroyed by spike protein disease. So, for instance, 70% of us are infected by those that were vaccinated. I'm one. I'm infected. I now have lupus, and I now have 3 different viral infections that are recurring on a rapid basis within my body because I've been infected by the vaccine. I don't know which one. I just know that mRNA vaccine got into my body from shedding and affected me, and it's the same results as if I'd been vaccinated. So when I tell patients about this stuff, I'm like, You know, this is what you have to do: dump iron, dump iron, dump iron, keep minimizing its toxicity if you've been vaccinated, because there's the toxicity. That's what is happening. It's attaching to beta 1 heme at the end terminal domain. I'm sorry, other doctors can't read or don't read. Mark Twain said it best: there's no difference. And so I know the world missed it, but they didn't understand iron, and they didn't understand its toxicity. And so that's really when you take it to that next level, that's when it all starts to make sense. We're like, dang,
Yeah.
this is this is this is really it. So everyone
So
focus
you know, I know I don't mean to belabor the point, but I get the upper limits of ferritin, but the lower limits I'm not there yet. So at what point, like, would you clinically, if you measured, would you take someone down to a ferritin of 0? I mean, is that even possible? And then would you say, oh, we need to keep going? And what would be your metric for deciding if we keep going? Like, in what frequency?
I love that question, and for lots of reasons. I'm a Christian, so I believe in Jesus. He's a pre-fall human, and I find it interesting that chlorophyll has magnesium within its hemoglobin molecule, and I suspect that our pre-fall human had magnesium within our chlorophyll. I'm sorry, within our heme molecules, which then wouldn't oxidize and we wouldn't die. Will God change that back? I don't know. Maybe I'm speculating and all that kind of stuff, but it's a very interesting concept: magnesium and hemoglobin versus iron, because iron oxidizes easily, right? Anyway, back to the question. I think that we um, I think we just have to realize that we have this C molecule as a gift, and we have to get everything fresh all the time to get C molecules. We don't want to be killing it. The grocery stores are killing it all. Everything you buy in the grocery store is killed because they're chilling it to 34 degrees. Remember, the maximum density water is 39.1. When you go below that, you get ice molecules. Ice molecules destroy the C molecule, literally cut it to pieces. That's why it tastes sour or bitter when you eat frozen. So we want to keep everything fresh all the time. And remember that when nobody's really done this right yet, I'm in the process of working on, and I have a corporate sponsor working on trying to get this C molecule isolated by a new mechanism. But the bottom line is, once we do that, then we're also going to get a whole bunch of stuff. We have a whole bunch of stuff planned. But the vitamin C lie will be my next book. I've been working on that probably 25 years altogether. And interestingly, Judith DeCava wrote her book in 1997, and on vitamins and trace elements. And she wrote the foreword for this book back in 2016. She's excited about it. So she's like carrying on that mantra, helping expose the fact of the harm of ascorbic acid being done worldwide.
You're familiar with the products? I think it's called Soma. It got kind of popular around in my circles 2 years ago, and there's an Indian uh, researcher that it basically was talking all about how we lost the ability to make our own vitamin C, and it was some type of catalyst for rebooting that system that's like dormant or lost or whatever. Is
No,
that something familiar to you?
that's bogus. You can't do that. And actually, that speaks to the one of the other amazing wrongs of science that's and conclusions have been going on for the past 120 years, no, 100 and well, anyway, it doesn't matter. Since 1906, it was when they discovered that primates, humans, guinea pigs, and bats that eat fruit don't make vitamin C. And they concluded that all the other animals that don't get scurvy made vitamin C. That was a wrong conclusion. It's still wrong to this day. Why is it wrong? Well, all those other animals eat other animals. They don't cook their food. They eat everything raw. Everything raw has vitamin C in it. Everything, even other animals. And um, some of this stuff is, I'm a hunter also. Some of the stuff you learn about how animals hunt and what they do is kind of fascinating. Like wolves, for instance, the alpha wolf eats the heart. Heart's the most nutrient dense of all the tissues. Nevertheless, it's really fascinating that we get so much recovery of our physiology from the C molecule on a regular basis. And back to the ferritin question, is the lowest ferritin level I've seen in a male was 1. I see a lot of young menstruating females that are not taking any ascorbic acid, and they'll have a ferritin levels below 10. And every other doctor everywhere wants to put them on iron. I'm like, no, you're fine. Oh, you've got plenty of iron. We can't measure it anyway. You're not anemic. Why do you need iron? And interestingly, Morley has exposed this. Almost all anemia is related to copper deficiency,
Right.
not iron. And so they're not doing the proper tests. So, and then they make the wrong conclusions. And then they think ferritin means iron. It doesn't. It's just a reactive protein that's only produced when you're not recycling. So if you're not making it, the level could be really, 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 really low, and that's really, really good.
So you would attribute like the symptoms of anemia to thyroid issues or to just mitochondrial dysfunction, low energy. So that because symptomatically it would be the same thing. So it's being conflated. You're saying essentially that like, oh, you might see like low iron markers, and the doc's going, oh, because you have iron deficiencies. Let me give you an iron infusion. And you're saying none of those iron markers are they're totally irrelevant because they don't really measure things. So your symptoms of anemia are really just a energy, like your lack of energy production. It's a metabolic problem.
It is, yeah. It's all related to the C molecule and copper almost every time. Um, I, I think that all iron infusions should be stopped immediately. They're harmful. Um, I find one of the most pitiful circumstances I see is chronic anemia, especially cancer patients that are put on iron. Or get, if I have cancer patients and they require a blood transfusion, I get them enrolled in hospice immediately because I know they're going to die. If they're a cancer patient and they have a ferritin level over 1,000, they're going to die. Get in hospice. There's nothing we can do. Um, and so the ferritin level is directly related to all death and disease, and it really makes a big difference of C molecules getting it down and iron, getting the iron out, keep reducing iron.
Well, I think if you're open to it, have to do a part two at some point because I feel like we've only scratched the surface, and this is fun. But to tie off what we've been talking about, the importance of potassium and what is your favorite source and what are your, daily amounts? What do you recommend?
That's another really, really good question because um, potassium is pretty plentiful in most of our root vegetables. There's only 4 vegetables that give you calcium. Those are broccoli, cabbage, kale, and cauliflower. And most people have an excess of calcium, so those aren't necessarily the best vegetables. Sometimes people refer to those as goitrogen type vegetables, but they're not. It's all related to calcium and lack of potassium. So remember, the most common form of hypothyroidism of all is lack of potassium. That's the most common. 90% of all hypothyroidism is just lack of potassium. And it's pandemic. The chances of people being hypothyroid, I mean, look around. How many people are above the ideal body weight? How many people have metabolic issues? It's so ubiquitous. It's unbelievable. It's not being treated properly. And interestingly, doctors don't understand the significance of T3 because it turns out that T3 thyroid hormone is the most active on the mitochondrial membrane. That's what controls the rate of exchange of potassium and hydrogen. So you have to have potassium for thyroid hormone to work. And you have to have T3 thyroid hormone. So all this mantra of give the patients T4 and they're fine, well, it fails 499 out of 500 times. I'm up to, like I said, almost 2,000 consecutive patients doing total reverse T3 ratios on synthetic T4. And it's consistently 1 in 500. I can't explain why 1 in 500 it works. I don't know, but okay, we just want to do the blood test. You don't need
So
any.
so root vegetables are a good source. Like potatoes are a good source. What's
Root
really a
vegetables
good
are really the key. And I found that I began blending about 3.5 years ago, 4 years ago now, blending my salads on a fairly regular basis. And after 18 months, I had the largest improvement in cholesterol numbers of any patient I'd ever seen with any treatment ever. After 36 months, it was even better. My HDL was up to 84 from started out at 42. My LDL went down, and my VLDL went from 44 to 11. I'm like, seriously, I looked at my results. I thought, dang, doctor, you need to get on some medicine or do something different, And I just started blending my salads, and I got those results, and I'm like, dang, that is so impressive. But it's so delicious once you learn your spices and you add to figure out how much water. What could be your favorite snack for the day to be sipping on? A salad with spices in it. It's so awesome. It's amazing. So you want to
coconut water and watermelon juice are also quite high in potassium.
yeah, but back to that original question. When my patients have a low intracellular potassium, I consider it a crisis. And I put them on potassium prescription. I put them on potassium rich diet. I put them on potassium supplements. And I'll tell them that, the maximum I can write for a prescription is 3 or 4 times a day. If I write for more than that, the pharmacy is going to report me to the medical board. I went in for my pre-op for when I broke my back, and the doctor asked me how much I was taking. I said, well, 12,000 to 16,000 milligrams of potassium daily. He looked at me and said, aren't you worried about your heart? I'm like, no, I'm really worried about my heart if I don't take it because I don't want to get atrial fibrillation. So all atrial fibrillation is related to this membrane potential problem from lack of potassium. So I was taking these large doses, and I told him, I said, well, I'm not worried about it at all because I know my intracellular potassium was down to 7.5% of normal, and I'm not taking it all at once. So as long as I didn't take it all at once, I know I'm going to keep accumulating potassium. After 6 months of doing that, it went up to from 7.5 to 15%. So did I achieve toxicity? Not by a long shot. It's not easy because remember, it takes ATP to attach the potassium. Am I struggling to get ATP? I could be. I don't know. This aging stuff has really kind of got me. When I broke my back, I all of a sudden started feeling aging, and I'm 70 years old, so I'm supposed to feel aging, but I'd never felt it before. Maybe I'm in denial.
Well, pretty good compared to most
You keep trying
70s.
to be healthy and keep doing healthy behaviors and things like that, and you kind of want to expect more results, but I'm a perfect walking, talking example that, there's always one more thing that you can learn. There's always one more thing that makes a difference. But make sure you get those minerals every place and get your hair test analysis. Find out what your potassium is. Find out what your ferritin is. Most important of all blood tests. The only blood test directly related to death and disease always wants to be below 100. If you're in the middle, that's terrible. You gotta be low. Low is better. And I know doctors don't know this stuff, and it's unfortunate because it's not wrong. I tell them now to their face, you can't make me wrong because you don't know what I'm talking about. Don't do that. Um,
Okay, so we got make sure you get potassium. You gave us some strategies for that. Get your iron down, your iron down. Use ferritin as a proxy for that, and then it sounds like whole food vitamin C is quite good. Something that they talk about quite a bit in the RCP. Are you a fan of acerola cherry and the sort of the adrenal cocktail, or what are your strategies for whole food vitamin C?
yeah, that's another good question. The problem is freeze drying destroys the C molecules. And almost all the products that are out there that don't have ascorbic acid that claim to be whole C, they all come from freeze dried powders. None of them work. None of them. None. Zero work. They don't work. They won't lower your ferritin. That's how you can find out if it's working. Your ferritin goes down. And so We have determined that the innate response C is still 98.5% intact C molecules, so it's useful. The half-life of the C molecule is 33 and a third day. The half-life of ascorbic acid is 90 minutes. So if you take one at a time of the vitamin C that I recommend, you end up with 18 hours a day of net benefit of C molecules.
So
I
you
think
said
that
innate response.
innate response is still the one I
Okay.
recommend. I went to that company and asked them to make it without the ascorbic acid, and they refused. But I'm like, no one else in the world was saying what I'm saying, and I'm like, would I listen to me if I was a major corporation making millions of dollars on the best possible seed product possibly on the market today? They added ascorbic acid to please the FDA, quite literally, and it violates their mantra from where they started in the big picture. But it's a company that who's trying.
And so, because it is a whole food source with the added ascorbic acid, are the negative effects of the ascorbic acid nullified because it's coupled?
Yeah, there's like I said, the half life is 90 minutes of the ascorbic acid. Yeah, there's some negative effect to the ascorbic acid, but it's going to be out of your body in less than 3 hours. So if you take one of the innate response at a time, you end up with a net result. Now, can you do that with fresh fruits and vegetables all the time? Maybe, maybe. How
And
about
what about dried fruit or juice?
that's a great question. Yeah, I interestingly, all the freezing is destroyed, so all my raspberries get dried now. I dry them on a Senesco dryer at about 100 degrees. Don't heat them, just dry them, and then I turn them into fruit leather. I found that blending shortens the drying time by about 3 times, so it's 1/3 as long. And then I cut them into little pie pieces because my dryer's round, and then I basically put them in Ziploc bags or vacuum pack them. Or sometimes I vacuum pack. Most of the time I just put them in Ziploc because it's dried, so that those same molecules are intact. They're going to last pretty much 1,000 years.
Amazing. Yeah,
I know it's
that's
amazing.
amazing.
Yeah, yeah, yeah.
So I like this strategy of making your own fruit leather. I mean, this is a traditional way of doing things. You think back to 150 years ago, people wanted to eat fruit. In the wintertime, they were making preserves, they were making fruit leather, they were taking the surplus of fruit that was occurring in the fall, in the summer, and they were preserving that. So this is a really cool traditional human strategy.
Yeah, it is, and unfortunately, we got more lazy because freezing is easier, it's faster. Throw them in a bag, vacuum pack them, whatever. But it's all destroyed. So I was like, when I can't realize what I was doing to all my raspberries, I used to pick, like about 15, 20 gallons a year, and of raspberries, and freeze them. And I don't do that anymore. I vacuum pack everything. I mean, I'm sorry, just dry everything and at room temperature basically, and then turn it up. And I find just fruit leather speeds it up. But you're right. I was amazed when I met a woman that was actually doing fruit leather, been doing fruit leather her pretty much her whole life, raising 6 children and all that kind of stuff. And I'm like, wow, I don't know why it is. Some people just figure this stuff out sooner than others, but
Yeah.
definitely everyone that's listening should begin to think about, preserving their fruit and vegetables with fruit leather and or drying at room temperature. Don't heat it. Don't kill it. Keep everything fresh all the time or dried. Great source if dried. All the nutrients are still in there when it's dried. It's just the water's come out, right?
Yep.
So,
Amazing. Tell us about your forthcoming book and where that can be found.
yeah, we're hoping this will be done by around the 1st of May. It's called the Calcium Life 3, mostly introducing these new principles of dilution and dipole zippers and the importance of potassium, a little bit about iron, but not a lot. But the next book is the vitamin C lie, explaining. I've been working on that for a long time, but that's going to take all this information with iron and the C molecules and put it in one place where everybody can, kind of like digest it a little bit more efficiently. And then the next book is the TSH Lies. So I call that the trilogy. Those three books are kind of pivotal to the change in the practice of medicine worldwide. Everything has to change. It's not okay for doctors to continue doing what they're doing. Today, it has to stop. It's not tomorrow, the next day. And it doesn't matter what their opinions are because it's basic science. They have to learn it. Otherwise, they're
Yeah.
harming patients, and that's not okay. First rule of medicine: do no harm. So I don't know how to make this happen any faster, except keep writing my books, keep teaching, and and we're going to do more of them after that. I have a book on nutrition and pregnancy, explaining makes such a difference, especially iodine, and I have a book on the superheroes of nutrition. I'm really excited about that. Explain some of the other supplements that I consider just like so fantabulous, like curcumin and rice bran, and just fantastic supplements from a nutritional standpoint. So I call it the superheroes of nutrition. Melatonin, another one, is just like a superhero. Most people don't know about melatonin. Have you done a show about melatonin?
I haven't done one about it, but yeah, it's pretty amazing, especially in the mitochondria and its production there.
Yeah, we could do a whole show on my on melatonin. Honestly, there's over a
Okay,
hundred health benefits, and
wonderful.
I I'm so impressed with melatonin. I just like crazy good. My cancer patients, I put them on a minimum of about 540 milligrams a day, and viral infections. I'll take melatonin 60 milligrams every hour for viral illness until I feel better.
Wow.
I know.
Whoa. Yeah, we got to talk about that. Okay. Well, and lastly, where can people find you if they want to learn more about you?
Probably the best places are website Aurora Health, like the Northern Lights, Aurora Health and Nutrition, all one word typed together, and or go to. We have some new websites coming up. Somebody stole my old website, thecalciumlie.com, and so it's not active anymore. But Aurora Health and Nutrition is probably the best place. You can order the books, you can order the hair tissue mineral analysis, you can order the minerals. There's a couple different types, and then it comes in liquid and tablets. But the electrolyte formulas that we sell have added potassium. That's all you got to know. The added potassium is correct and corrective for 90% of us. The other one is correct for everybody, but it's not corrective for those that need potassium. But it doesn't hurt you, so it's all good. So those people that have added potassium that need to know, at high potassium need to know that's out there that happens. It's typically associated with stress. And we find that the heritage muscles might be one of the most discerning aspects of physiologic stress that we have. My sweetheart's a PhD psychologist, and she's using heritage analysis on all her patients now, and she's beginning to recognize who has significant physiological stress based on their heritage analysis, because they will first retain, this is chapter 8 of my book, they'll first retain sodium and then potassium and then, interestingly, lithium or rubidium, and that generates the resistance of the type 6 hypothyroidism from the accumulation of lithium or rubidium, which you can only diagnose with hair tissue mineral analysis. But it's fascinating, and it's all reported in the literature, but no one's paid attention to it.
Well,
So,
Doc, thank you so much. This was really enlightening and fun.
hey, thanks. It's been my privilege always to teach.